This video is going to take us through Lab 10, which covers human genetics. To start us out, we need to talk a little bit about pedigrees. It's mentioned in that first chapter, but it's not really discussed in detail until later on. Pedigrees are essentially just family trees that look at a particular genetic disease. We use pedigrees because we can't just breed people together to see whether or not a trait is genetic, and if it's genetic, how does it get inherited. Instead, we have to look at past matings to try to figure out how that trait might have gotten passed down from one generation to the next. We're going to talk later on about how to read these things, but this is how we essentially study human genetics, since we can't just do breeding experiments with humans. After that, they talk about some traits that there's some evidence that there's genetic components to. They start out by handedness, right-handed or left-handedness. What I want to mention is this is, again, vastly oversimplified. People can be taught to use either hand. So left-handedness is actually more common in the population. It's just that most school teachers prefer for students to be right-handed. It makes it easier for them to teach them. So just keep in mind, this one is also, there's a huge environmental component to this one. So what they tell us in the scenario in the lab manual is that being right-handed is a dominant condition, being left-handed is recessive. They give us this little problem of Kenneth. Let's try to figure out what Kenneth is. He's right-handed, but his father is left-handed. With that information, can we figure out exactly what his genotype would be? This is where we have to remember what genotype and phenotype actually mean from the last chapter. So genotype is looking for the actual genetic makeup, and so we need to figure out what his R's are. So if the father is left-handed and left-handed is recessive, that means we know the father must be little r, so we can fill him in the pedigree. We can go ahead and bring the R's into the boxes that are down below them. Then what we know is that Kenneth is right-handed, so he must have at least one capital R. We can put it right here. That would make this box right here big R, little r. What that means is, since this box is showing us what Kenneth must be, in order for him to be right-handed, he's got to have the big R that he must have gotten from mom, but because dad only has little r's to give him, that must mean that he's heterozygous, and so Kenneth would be big R, little r. Next, they give us another scenario. Beth is right-handed and both of her parents are right-handed as well, so what is her genotype? Okay. We don't know a whole heck of a lot about this particular problem because we don't know if her parents are homozygous or heterozygous. We don't know if both of her parents are this, both of her parents are this, or one parent is this and one parent is this. All we know, really, is that Beth is right-handed. If we can't tell from the problem that we are given whether or not a person, or an animal for that matter, is homozygous or heterozygous, the way that we write that is we give them what we know they must have. We know she must have one big R because she's right-handed, but we don't know what her second letter is because she might be big R, but she might also be big R, little r. So if we're unsure, this is how we write it. The second letter gets a dash to indicate that we're uncertain what their second allele is going to be. Next, they get personal and they ask you about you. What hand do you write with, and therefore what is your genotype? I'm going to use me as an example because I don't know what you guys write with. I happen to be right-handed, so I would answer right. And then what's my genotype? I know I've got a big R, but I don't know what my second letter is. If you happen to be left-handed, you would say you're left-handed, and then you do know what you are, because in order for you to be left-handed, you must be homozygous recessive, so little r, little r. Next. The clasping the hands thing. This, again, is going to have some environmental components to it. What I want you to do is, without thinking about it, just clasp your hands like you see in the picture. When you put your hands together, one of the thumbs ends up being on top. The left thumb being on top is dominant. The right thumb being on top is the recessive condition. What they ask you next is, you personally, which thumb is on top for you? Now, I happen to do just like this picture does up over here, so my right thumb ends up being on top, which means I do know exactly what my genotype is. I'm little t, little t. If, on the other hand, your left thumb ends up being on top, then you just know one of your letters and you don't know what your other letter is, so you're big T and then a dash since you don't know the second one. Earlobes. There's multiple options for earlobes, and of course there's environmental components with people putting those stretcher things in their ears to stretch their earlobes out. But the two options that we are going to talk about that there is a known genetic component to is what I'm showing you in this picture. You can have the free earlobe, which is also called detached sometimes, or you can have the attached earlobe, which just connects straight down and it doesn't go back up like the free earlobe did on that side. Having the free earlobes like this is the dominant condition. If your earlobe looks like that, you're big L dash. If you have the attached earlobe, then you know you're little l, because that's the recessive condition. So think about what your phenotype is, and then based on that, determine what your genotype is. Just as a side note, neither of these factors matter. You are not more or less likely to get any diseases based on where your earlobe happens to be. And most people don't even notice earlobes, so it's nothing depending on which one of these you has. It doesn't mean anything. Freckles are what we have next. Having freckles is the dominant condition. She has freckles, and so we know she would be big F dash. Not having freckles anywhere on your body means that you would be little f, little f. So if you need to look over your whole entire body to see whether or not you have a freckle anywhere, a freckle is not raised like a mole is. But if you have even one freckle someplace on your body, you would have at least one big F. If you are completely lacking freckles, you're little f, little f. Next, we're going to talk about chromosomes for just a second again, because remember, your genes are carried on your chromosomes. This picture is what's known as a karyotype. It's a picture of all the chromosomes that can be present inside of a cell. This happens to be an abnormal karyotype, and I'll talk about why it's abnormal here in just a second. Most of your chromosomes that are numbered 1 through 22 in this picture, they code for everything from how to make your insulin and apparently where to put your thumb when you clasp your hands. Those chromosomes are called autosomes. But this remaining chromosome set off down here, they determine what sex you're going to be, plus some other things. Because they determine the sex of an individual, they're called sex chromosomes. The two normal options for sex chromosomes are XX is female and XY is male. And you can tell the difference between X's and Y's because the X chromosome, like this one right here, is very big. The Y chromosome is a very small little chromosome. That's why I usually write the Y as a lowercase y, because it's a tiny little nub of a chromosome that essentially just says, here's how you make testosterone and a testosterone receptor. There's not a lot of other things that are carried on that Y chromosome. So the questions that it asks you are, what's your phenotype? In other words, from a genital/chromosome perspective, are you male or female? I am not looking for your gender identity. I just want your biological sex. So I am a cis female. I would put that I'm a female. What is my genotype? Because I'm female, I would put XX. If I were a cis male, I would say that my genotype is XY, but I am not a cis male. Transgender individuals, it's different. Your biological sex would no longer match your genitalia, but your chromosomes don't change when you do a sex reassignment surgery. Now here's where I want to talk about a little bit about sex versus gender. Sex is a biological construct that is generated by your chromosomes plus your hormones that help develop the genitalia that will make you externally match the sex that in theory matches your internal chromosomes. That said, sometimes hormones and chromosomes have conflicting information in them, and as a result, a person might chromosomally be a female, but because they produced more testosterone than they were supposed to, maybe because of a tumor or something along those lines, they end up, the clitoris grows enlarged and it looks like a penis, their ovaries may drop and develop more like testes, and they might end up looking like the opposite sex. The term for a baby that is born that looks sort of like a hermaphrodite is intersex or middle sex. What's extra creepy about that is, especially pre-karyotyping and other things, if a child was born intersex where they weren't sure what the sex was, which happens way more often than you might think, the parents would be asked to choose, what sex do you want this baby to be? And then they would do a surgery to make that child that sex. And sometimes the parents chose wrong, and the sex did not match the chromosomes. And so the child's gender identity didn't match their genitalia identity as a result of a surgery that happened before the child was even aware that there was a problem going on. So what I really want you to understand here is gender is not as simple. It's not a binary thing. There are things that go wrong when it comes to chromosomes and hormones. Now let's come back to this pedigree. These are the normal sex chromosomes that can happen. However, the person that produced this karyotype is XXY. They have two X chromosomes and a Y chromosome. So if you were looking at this and you had to determine the sex of this person, what would you call them? They have two X chromosomes, that's female, but they also have an X and a Y, and that's male. Well, what I will tell you is, ordinarily, as a person who is XXY starts the process of development, because they have a Y chromosome, they will end up developing male genitalia, and in fact can live a normal life as a male and even reproduce and have children later on in their lives. And so this is one of those cases that can end up being confusing, so that you hopefully understand gender is not black and white. It's not binary. And so there is a reason why there is a movement to try to understand more about gender misidentity or gender nonconformity, and it's because biology is never as simple as some people try to make it out to be, including the lab manual in this case. Okay, with that said, they want us to just do a basic Punnett square that is a cis male with a cis female. And so we'll put the female up here because sometimes she likes to be on top. And we'll put the male off down here. As we fill it in, just like any other Punnett square, bring the x's down, bring the xy's over. What you will see is, statistically, from a probability standpoint, anytime two people have sex, there's a 50-50 chance that the child is either male or female. The other thing that I want you to understand about this is, because women only have X's to give, every single one of her eggs has an X chromosome, the person who determines the sex of the baby is actually the male. Because half of his sperm carry an X chromosome, the other half of his sperm carry a Y chromosome. Now, once again, disclaimer and reality check. There are plenty of families that are out there that have had 12 kids and all 12 of them are males, and none of the females have been carried to term, where they never even had a female. So just because there's a 50-50 chance doesn't mean that's necessarily what's going to happen in your family. If, however, you look worldwide, probability and averages tell us that 50-50 chance you have a male and a female. Okay. Now, I mentioned earlier the X chromosome is a longer chromosome than the Y chromosome. That means it carries more genes than just, here's how you make ovaries. It carries genes for blood clotting factors. It carries genes for some of the proteins that are in your eye that help you see color. So there's a bunch of other things that are carried on those X chromosomes. Anytime you have a gene that's carried on either of the sex chromosomes, it's called sex-linked. But because the X chromosome is bigger, sex-linked usually means X-linked. When you're solving a sex-linked problem, you can't just say like big A, little a. You have to write your code on the sex chromosomes, because the sex of the individual will determine the outcome of a potential cross. So the problem that we're going to work to start us out with that is about color blindness. I mentioned that some of the genes on the X chromosome code for proteins in the eye. Well, some of those proteins can be found in cones that help people see colors. They've written us a code here. I'm not a huge fan of the way they write their code, but if it works for you, fantastic. But here's the thing. If you have normal color vision, that big N means normal vision, but you have to write it on the X, because that N is literally carried on the X chromosome. Color blindness is the recessive condition, so it gets a lowercase n, and I usually write it, to make it easier for me to read, down on the X instead of up as a superscript, just to remind myself it's a lowercase recessive condition. So they want us to cross a colorblind male to an XN female. Okay, so X, big N, X, big N. That's the female. They told us that. We're crossing her with a colorblind male. So he's male. He must be XY. He's colorblind, so he's going to get a lowercase n. The Y does not get an N because that gene is not located on the Y chromosome. It's only located on the X chromosome. That's why you have to consider the X's and the Y's, and you have to write the code on the X's because it's carried on the X. From here, you still just carry everything down and over, like always. So that goes there. That goes there. Bring the side stuff over. And then answer whatever questions they have. What is the genotype of the colorblind male? That's what I wrote right here, but I'll write it again. X with a little n, Y. What proportion of the kids will be colorblind? As you look at this, if a person has at least one capital N, they can see color. So that has normal vision. She has normal vision. He has normal vision. He has normal vision. So 0% of their kids will be colorblind. What proportion of the daughters will be colorblind? Well, if none of them are, then none of the daughters are. And if none of them are, none of the sons are either. So if mom is completely normal, all of the kids are going to be phenotypically normal. They're going to be able to see color. Next, we're crossing a colorblind male to a carrier female. So a carrier female is one that can see color, but does carry that colorblind gene and can pass it on to her offspring. She's hooking up with a dude who is colorblind, so he looks like that. Bring stuff down and over as per usual. My messy handwriting is also one of the reasons why I like to make my lowercase n's at the bottom of the X. I really can't screw it up if I write it down there. Okay. What is the genotype of the carrier female? Again, X big N. Go away. X little n. What proportion of the kids are going to be colorblind? Okay. She is a normal carrier. She is colorblind. He is a normal carrier. He is colorblind. That means 50-50 shot. So 50% of those kids are going to be colorblind. What proportion of the daughters are colorblind? When they ask for just the daughters, ignore the men. So these are the ones that are daughters. 50% can be colorblind. What proportion of the sons? These are the sons. 50% of those can be colorblind. This time, normal vision male, so X big N, Y. This time I'm going to put the guy on top because, again, sometimes you got to flip things around. He's being crossed to a carrier female, so X big N, X little n. Bring your stuff down and over. What is the genotype of a normal visioned male? It's what I wrote right there. What proportion of the kids are going to be colorblind? So normal, 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 colorblind. So that's 1 out of 4 or 25%. So 25% chance they make a colorblind kid. What proportion of just the daughters? Well, this time our daughters are on this side, and neither of those daughters are colorblind, so 0% chance they make a colorblind daughter. What proportion of the sons? These are our sons, and 1 out of 2 of those can be colorblind, so 50% chance if they have a boy, that boy's going to be colorblind. She does provide you the hint that I was kind of going over as we do that. Make sure as you read the question, you read what it says. Like if they just ask for the sons, don't consider all the kids, just look at the sons. All right. After that, we're going to come back around to pedigrees. I mentioned that they're family trees that look at a particular trait. Stuff that you need to know about a pedigree. Anytime you see a circle, that means it's a female. Incidentally, the abbreviation for a female is that. Anytime you see a square, it's a male. Incidentally, the abbreviation for a male is that. If you have a female and a male that are hooked up with a line that goes straight across like that, we're going to be euphemistic and say that that means that they're married. Really, it just means they had sex and made babies, but we're going to pretend that it means married. If there's a line that goes up and over to connect individuals, that means that they're siblings. In this case, 2 sisters. Well, 3 sisters. There's another one off over there on the side, but up and over, siblings. So, looking at this, when you have either a box or a circle that is shaded in, it means they are affected by whatever the condition is that you happen to be looking at, whether it's color blindedness or albinism or muscular dystrophy. Doesn't matter. If they're shaded in, they have the disease. If they're not shaded in, they are unaffected or don't have the disease. Next, off to the side, Roman numerals indicate the generation. And so this, if you kind of wanted to go back to Mendel's terms, that would be the P generation. This would be the F1 generation. Then we use Arabic numerals, the 1 and the 2 here, to indicate the person within that generation. That allows us to talk about a person without assigning a name to them. So if I were to ask you something like individual 2-4, what sex are they? Well, you come down to 2 and then over to 4, and that's a male because that's a box. And so those numbers just help us identify the people so we can talk about them. Okay. There are 3 different inheritance patterns that we expect you to be able to recognize when you're looking at pedigrees: dominant, recessive, and sex-linked. We're going to start by talking about recessive. If you look at a recessive pedigree, the trait that you're looking at or the disease that you're looking at tends to skip generations. So notice nobody in generation 1 had the disease, but somebody in generation 2 did. The other thing that you need to look for is that you have a kid who is born to unaffected parents. And so this kid has the disease, but neither of the 2 parents do. That can only mean that you have a recessive condition going on. And so that's what you're going to do to recognize how that pedigree or how that disease is getting inherited. Okay, here's an example that we're going to look at when it comes to recessive conditions. Albinism. We talked about this last week with the peacock. It can happen in people too. The person can't make melanin, and so they end up being super duper pale. What it says is blah blah blah. So normal pigmentation is dominant, albinism is recessive. Now we know that it's recessive because as we look at the pedigree, we skipped the first generation and it showed up in the second, and we have a kid who was born to unaffected parents. So albinism is recessive. The pedigree tells us that. Individual 2-4 has albinism. We know that because they're shaded in. Notice that neither individual 1-1 or 1-2 is affected, but individual 2-4 is. How does that happen? Well, what happened is, since this is a recessive condition, both parents can be carriers for that condition. So, what is the genotype of individual 2-4? In order for them to be an albino, which is recessive, they must have lowercase a, lowercase a. If they have lowercase a, that means both of the parents must have had lowercase a's, but they are both unaffected, which means they also must both have capital A's. And so we could fill in the pedigree, or pardon me, the Punnett square from that point. That is already there. And that is already there. So, what are the genotypes of parents 1-1 and 1-2? They're both heterozygous carriers for albinism. Next, dominant pedigree. When you see a dominant pedigree, it's going to pop up in every generation. And when you look at a set of parents, about half of their kids are going to end up with the condition. And notice I said about. It doesn't have to be exactly 50-50. Especially because, like as we look at this set right here, these parents only had 3 kids. You can't really do half of a kid having a thing. So it only has to be an approximate sort of a situation. So, for this one, we're going to look at Huntington's chorea, aka Huntington's disease. I don't know if you guys remember the show House from back when it was on, but there was a whole story arc about 13, who was played by Olivia Wilde, not knowing whether or not she had Huntington's disease. Her mother had died from it, and did she want to know if she had it, and all that sort of jazz. This is the disease that they were talking about in that show at that time. Okay, so let's look at this. If individuals 2-5 and 2-6, that's this person and this person, have another kid, what's the chance that their next kid will have Huntington's disease? Okay, here's what we got to figure out. First off, is Huntington's disease dominant or recessive? As we look at the pedigree, it's showing up in every generation. About half the kids of a parent have it, so it's dominant, which means this time capital H means Huntington's, not normal, like it did over in albinism's. Lowercase h means normal. So for everybody who is unshaded, you know they're lowercase h, because the only way to be normal is to be homozygous recessive. Now that we know that much, okay, so we know this person is that, this person is that. Well, in order for this person to make kids that have it, we know that both of these kids must be heterozygous. And again, since this person had a parent who was homozygous recessive, we know that that parent must be heterozygous as well. What is the genotype of individual 2-6? Okay, so 2-6, we figured them out earlier. They're lowercase h, lowercase h. We know that because they're not shaded in, and not shaded in is normal. What is the genotype of individual 2-5? Well, we figured them out earlier. They're big H, little h. And that we know that because they had a parent that was unaffected, but they do have the disease, so they must be heterozygous for that disease. Now, the original question was, what's the probability that they have a kid who has Huntington's disease? We would have to do the Punnett square with him and her to figure out probabilities. And as we look at my messy little Punnett square, these two kids have Huntington's disease. These two kids are normal. So it's a 50-50 shot every time they have a kid that their kid might end up with the disease. What is the probability that 2-7 will have Huntington's? Well, that's what we just answered. I guess I just asked that question twice. All right, pardon me. Next up, sex-linked pedigrees. So sex-linked diseases are usually sex-linked recessive diseases. So yes, we're going to skip generations and have kids that are born to parents that are unaffected. But more importantly than that, as you look at this pedigree, I want you to notice it's exclusively males that are affected by the disease. Now, it's possible for females to get it. It's less likely for females to get it. And so when you look at a pedigree and it's almost all males that have it and just a couple females, that's going to be a sex-linked condition. Remember, anytime you see sex-linked condition, you have to think about the sex chromosomes as you're solving the problem. So, this time they give us the code up here. X with a big D is normal muscle tissue, because remember, this is a recessive condition, so the little d means that a person can have muscular dystrophy. The Y chromosome doesn't get the D's because it's not on the Y chromosome. It says to cross a normal male is going to be X with a big D, Y. To a carrier female, she's going to be X with a big D, and then X with a little d. Fill it in as per usual. And then answer whatever questions they have. How many of their sons are predicted to be affected? So these are the sons. This person's normal. This person has muscular dystrophy. So 50% of the sons can end up with muscular dystrophy. That's a 50%, not 500. What is the genotype of a normal male? If they're normal, they must have a big D and then Y for a male. What is the genotype of a carrier female? X with a big D, X with a little d. How many of their daughters are predicted to be affected? So this is our daughters. She's a carrier. Oh wait, no, she's not even a carrier. That's a big D. This is why I write my stuff on top. She's completely normal. She's a carrier. But neither of them have muscular dystrophy, so 0%. Why are more males affected by sex-linked traits than females? Okay, so here's what you have to keep in mind. Females have 2 chances to get a good copy of a gene because they get 2 X chromosomes. Males, on the other hand, they only get 1 chance to get a good copy of that gene. So that's why males tend to be affected more often by sex-linked disease. They only get 1 chance to get the right gene. All right, after that, we have questions that are going to go over everything that we just did. So right-handed is dominant over left-handed. Cross 2 people who are heterozygous for the right-handed allele. So big R, little r, big R, little r. What proportion of the children are going to be left-handed? That's 1 out of 4, which I'm perfectly happy if you write 1 out of 4, or if you want to turn that into percent, you can. 25% are going to be left-handed. In interlocking thumbs, the left thumb on top is dominant. Jorge's left thumb is on top, but his father's right thumb is on top. So that means his father I think we use T's for this. So his father is little t, little t. What is Jorge's genotype? Well, in order for him to put his left thumb on top, which is the dominant letter, he must have a big T. Since his father only had little t's to give, that must mean he's heterozygous. Kiara has attached earlobes, little l. Both of her parents have free earlobes. How is that possible? What are her parents' genotypes? Well, this kind of goes back to that carrier sort of situation that we were talking about. So if she's little l, that means each of her parents must have a little l, but her parents both have free earlobes, so that just means both of her parents must be heterozygotes in order to make her off over there. There's only a 25% chance they made her, but they made her, so both of them are heterozygous. Jeff has freckles, but neither of his parents do. Is that genetically possible? Okay, so remember that not having freckles is the recessive condition. So is it possible that they made him with his big F? No. This is one of those tricky sort of situations again. Maybe they adopted him, maybe he was swapped at the hospital, maybe they used some sort of artificial reproductive technology to create him, maybe he had a spontaneous mutation. There's lots of reasons why he may end up having freckles while his parents didn't, but assuming no mutation and normal other stuff, no, he's not their kid. Okay. Hemophilia is a sex-linked recessive disease where a person is missing a clotting factor and their blood doesn't clot very quickly, so they can bleed to death from even a very small wound. Normal blood clotting is dominant. They've already kind of screwed this up because they made that a B and the other one an H, and so we're going to change this to a capital H so that we can keep our letters the same. Cross a carrier female to a normal male. So what's the genotype of a carrier female? X big H, X little h. What's the genotype of a normal male? X big H, Y. From there we can put those into our Punnett square. And what portion of the children can clot blood? Okay, so this person can clot, this person can clot, this person can clot, this person's having a bad day. So, 3 out of 4, which again I'm perfectly happy with you giving me a fraction, that is the same thing as 75%. What proportion of just the girls can clot? These are my girls, both of them can clot, so 100%. What proportion of the boys? These are the boys, 1 out of 2, or 50% can clot. Use the pedigree above to answer the following questions. Okay, so this time they're not telling us how the disease is getting passed down. We have to look at the pedigree and figure it out for ourselves. So first thing you need to notice is how many generations is the disease popping up in? And this one's doing 2 out of 4, so it's only popping up in half the generations. If it skips generations, it's going to probably be one of our recessive conditions, either just straight autosomal recessive or sex-linked recessive. So next we need to look at, is it affecting males and females at the same rate? And yes, it is. So that means this is a recessive condition. Which disease could produce this type of pedigree? Huntington's, muscular dystrophy, or albinism? Well, Huntington's was a dominant condition. Muscular dystrophy was sex-linked. Albinism was our example of a nice autosomal recessive condition. So since this can be albinism, I'm going to use A's to indicate everything here. Now, since we said right here that this is a recessive condition, lowercase a equals albino, which I'm spelling wrong. Capital A means normal. If a person is shaded in this time because it's a recessive condition, they must have little a, little a. So we can fill in both of those people, and then we're going to start extrapolating from there. But let's come down to the questions and see what it asks. What is the genotype of individual 4-2? Here, look at that. That's one that I just answered, because that's this person, so they're little a, little a. What's the genotype of individual 3-3? Okay, that's this person who happens to be the father of this person. They are normal because they're not shaded in, so we know they get a capital A. They produced this kid that has two little a's. One of those little a's must have come from 3-3, so they are heterozygous carriers for albinism. What is the genotype of individual 3-4? Well, that's this mother over here. She also contributed to this daughter going on over here. She is also normal because she's not shaded in, but she made a child who has albinism, and so she is also a carrier. Are individuals 3-3 and 3-4 related? Okay, well, there's a couple ways we can answer this. First off, they're married to each other, or at least they're having sex with each other, so there's a husband and wife thing. But I want you to take this a step further. And as you look up, so these are his parents and his grandparents. These are her parents. Notice that she has a mom who is related to his mom, and she has the same grandparents up there. That means not only are they married, but they're cousins. So yes, they're married cousins. Some countries consider that to be incest, some don't. Kind of depends on which country you're visiting at the time. But what I would like for you guys to notice is when you have incestuous relationships like cousins marrying, you are more likely to have diseases pop up in the population. That's why there tends to be taboos on incest. It's because diseases are more likely to pop up. Use the pedigree above to answer the following questions. How is this inherited? So as you look at it straight away, you can see the diseases popping up in every single generation. You can see that it's affecting both males and females. That crosses sex-linked off the table. If a kid has it, every single time one of the parents has it, so that means this is a dominant disease. Which disease could produce this type of pedigree? Huntington's is a dominant disease. So we're going to use H's just so that we have our code going on. Since we said it's dominant, H equals Huntington's. Big H equals Huntington's. Little h is normal. This is where people tend to screw up the most often when they're looking at dominant pedigrees. They always assume the lowercase letter is going to be the disease. The whole point of this pedigree is it showed you this disease is dominant, not recessive, and so that means the uppercase letter indicates the disease. Any dominant condition, the uppercase letter indicates the disease. What is the genotype of individual 1-2? 1-2 is this person. They are normal, not shaded in. Normal is the recessive condition. That means they are lowercase h, lowercase h. What is the genotype of individual 1-1? Okay, we know they have a capital H because they are shaded in. As you look at some of the kids that they produce, some of their kids aren't shaded in, which means they're lowercase h, lowercase h. So that means dad must be a heterozygous person. He is not a carrier. He has the disease, and there's a 50% chance anytime he has a kid, that kid is going to have the disease. We're going to look at that in the next question. If they have more kids, what's the probability that the next child is affected? Apparently they did not give us a pedigree, or a Punnett square. I keep doing that. This time, so we'll do the 1. Put dad up here, mom down here, fill it in. And as you fill it in, you will notice these kids have Huntington's, these don't. So anytime they have kids, 50% chance that kid is going to end up with Huntington's disease. Use the pedigree to answer the following questions. How is it inherited? It is skipping generations. That tells us it's recessive. But it also is only affecting males, not females. So that means not only is it recessive, it is sex-linked recessive. Muscular dystrophy was our example of a problem that worked that way in a sex-linked condition. So did color blindness and hemophilia. They work that way too. Okay, since we were doing muscular dystrophy, we're going to make our letters D's. So since it is sex-linked and recessive, X with a big D is normal. X with a little d is muscular dystrophy, and then Y's are Y's. What is the genotype of individual 2-2? That's this male right here. He's affected, so he has a lowercase d, and he's a male, so he's got a Y. What is the genotype of individual 1-1? He's a male, so we know he's XY, and he's not affected, which means he has an uppercase D. What is the genotype of individual 1-2? Her. We know she's XX because she's a her. We know she has a big D because she is not shaded in. We also know she made this kid who had a lowercase d that didn't come from dad, so it must have come from her, so she must be a carrier for muscular dystrophy. So if he and her have another kid, what's the chance it's affected? So we put him here, and carrier mom here, and fill it in. Okay, so normal, normal, normal carrier, diseased. So more children doesn't care about the sex, 25% chance they have a kid with muscular dystrophy. What's the probability that the next son is affected? These are the sons, not affected, affected, so 50-50 shot when it comes to boys. What is the probability that the next daughter, that did say unaffected, but the same answer applies there. What is the probability that the next daughter is unaffected? Well, she's unaffected, so 100% chance they have unaffected daughters. And you know what? Now I need to come back up here because this is, again, goes back to make sure you read the question. I didn't do a very good job of that. It asked for children that are unaffected, and I had answered this as affected. So it's not 25%. The chance that we have a kid who's unaffected, that's 75%. Sorry, my screen got all fritzy. 75%. So notice how easy it is to not read the question really well and so answer it wrong because you assumed it was asking something, and just try to pay attention to that and don't make mistakes like I did. At least I caught the mistake before I posted my video, I guess. So yay! Just make sure you read those questions. I think that's the last of it, yes. So at this point, you should be ready to go and complete your post-lab quiz.